We will speak about visual analyzer first. Peripheral part of this analyzer receptors are located in an eye. We'll draw an eye. So we have the cornea, we have lens, and we have vitreous body that are transparent to light. So therefore, when an object is projected through cornea and lens, in normal eye, a sharp image reversed up bottom should be produced on the retina, and to see the objects at the different distance from an eye, the lens should change its shape, and to see close objects, ciliary muscle should contract so that the lens became more convex. And when the far objects, distant objects are observed, ciliary muscle relaxes and the lens became less convex, and this is called accommodation. So, but it not always happens so. There may be certain disturbances of vision. For example, myopia or short-sightedness. In this case, the object is focused not on the retina, but due to the elongation of an eye, the object is sharp, is focused before the retina, and on the retina the object became vague, not sharp. So person sees bad on the long distances. To improve the vision for such situation, it is possible to use biconcave lens. It will elongate the focus distance and will shift the sharp image on the retina. Another case is when person sees bad the close objects, for example the book. In this case, the object should be sharp, also not on the retina, but behind the retina, like that. So the sharp object is behind, on the retina it is vague. To improve the vision for such situation, to read or to see close objects, biconvex lens is used that decreases the focus distance and produces the good image on the retina of such eye. This is called farsightedness. So farsightedness or hypermetropia. And another type is called astigmatism. Astigmatism. is when the eye does not focus light evenly on the retina, and this results in blurred vision at all distances. And now we will speak about the structure of a retina that consists of light-sensitive cells. On this picture we can see rods and cones. They are placed on the peripheral part of the retina. Then we may see bipolar cells that connect one or several rods and cones with one ganglion cell, red ganglion cell. And ganglion cells, they produce optic nerve that carries the impulses to the brain cortex. We have three types of cones for color vision. We have cones for R, G, and B, red, green, and blue color. By combining these colors, the eye may see by mixing these colors, we may see many billions of colors of surrounding objects. When light reaches rods and cones, then they produce not action potential, but instead of it, they are hyperpolarized. This is the peculiarity of the vision. We have therefore receptive fields. At the center of an eye, one rod or one cone is connected with one bipolar cell and with one ganglion cell. On the periphery, we have larger receptive fields. We have many rods and cones that may be connected with bipolar cell, and therefore the receptive fields are larger at the periphery. At the center of an eye, we have more cones and less rods. On the periphery, the number of rods increases, of cones decreases. So therefore the peripheral part of retina is more light sensitive than the center part. At the place where optic nerve is produced, we have blind spot, but we do not feel it normally because we have binocular vision, and due to this binocular vision we have stereoscopic impression. We may estimate the distance between us and an object. It is very important. And also we have different position of blind spots in right and left eye, and therefore it doesn't decreasing and doesn't make our vision worse. To see well, an eye should constantly produce tiny and very rapid movements, secondary movements, and without these changes of the position of an eyeball, we will not be able to see. So the important condition to see well is that eye should constantly move with special muscles. An important mechanism is accommodation of an eye, which is produced by the lens. There is special muscle, ciliary muscle. When this ciliary muscle contracts, contracts, then lens became more convex, and an eye may see close objects. For example, during reading, when ciliary muscle relaxes, then the shape of the lens is less convex, and we may see far objects. So again, this is called an accommodation of an eye. Let's see how the information is processed in the brain. The second part of an analyzer is the conducting pathways. So it is produced, this pathway is produced by optic nerve, then optic chiasm, and optic tract after the chiasm. It is interesting to mention that the temporal retina is projected in the same hemisphere, and the information from nasal retina crosses in the optic chiasm the hemisphere and is projected in the cortex of the opposite hemisphere. And it should be mentioned that the information is processed in the thalamus, in lateral geniculate body. This is the center or subcortical center of vision, and from here, and the axons of the thalamic neurons in the thalamocortical tract in optic radiations reach the primary visual cortex, the 17th field in the occipital brain cortex, where the picture that is that it produces the image in our brain is developed. For analysis of the visual analyzer, we may estimate the visual acuity of vision, how sharp, how good we may see. And another type of analysis is the analysis of visual fields. So here we may see the visual field of the right eye. So for the right eye, we may see that the lateral side, temporal side of the visual field is greater than the nasal side of the visual field. And we may also see that there is the point of the sharpest vision in the center, and there is a blind spot here. And it also should be mentioned that the dimensions of the color visual fields are less, their dimension is less than the dimensions of black and white visual fields. Now we will speak about the acoustic analyzer. We have external ear, auricle, external acoustic meatus, and tympanic membrane. We have middle ear, so we have outer ear, middle ear. After the tympanic membrane, we have bones, malleus, incus, stapes. And we have inner ear that is in the temporal part of the skull, located in the temporal bone. And when we have the noise, then mechanical oscillations are conducted through external acoustic meatus normally to the tympanic membrane, and membrane conducts these oscillations to the oval window under stapes. Here we have in the center oval window, and this oval window produces oscillations of the fluid in the cochlea. So here we may see the oval window in the left upper corner, oval window, and the fluid path is going along the cochlea. Cochlea produces 2.5 turns, and then back in the cochlear partition, and just to the round window. And here, in the cross section of this cochlea, we may see the tectorial membrane and the organ of Corti. And here we have the tiny hair cells, mechanoreceptors, that are stimulated by the oscillations of the fluid in the cochlea. So what we have again here, we have tympanic canal, and we have vestibular canal, tympanic canal, and cochlear duct. And here we have the organ of Corti on the basilar membrane. This is an enlarged picture of the organ of Corti. We have hair cells here, and the special hair cells are mechanoreceptors, which do produce the acoustic nerve. And the different frequencies of oscillations do stimulate the hair cells in the different parts of the cochlea. It is shown here that at the base of the cochlea we have the hair cells that are stimulated by high frequencies, but high frequency like 20,000 hertz, high sounds, and low sounds, bass, low sounds like 200 hertz or even less. These sounds produce vibrations, produce stimulation of the hair cells at the apex of the cochlea, at the apex. So therefore, by such distribution of the hair cells that are responsible for different frequencies, we may differentiate the sounds of different frequency. The range of audibility of the human ear is shown here. So the lowest frequency is about 20 or 50 hertz at the left part of the graph, and the highest frequency that ear can hear is 2000 hertz at the right part of the scale. The threshold is different for different frequencies. The lowest threshold and the highest sensitivity is in the speech region. The speech region is about 100 hertz and up to 7,000–8,000 hertz female voices, and male voices are lower frequency. And we have also the threshold of pain, that is about 120 decibels. The sensitivity of the ear to different frequencies may be estimated during audiometry, special examination of the hearing analyzer, acoustic analyzer. Conducting pathways and the central part of the acoustic analyzer. So we may see here the cochlea and auditory nerve that is produced from the cochlea. Then we may look at the propagation of the impulses in the hindbrain, cochlear nuclei, then inferior colliculus, in the midbrain, and then medial geniculate nucleus in the thalamus. Lateral geniculate nucleus was for the visual analyzer, and here we have medial geniculate nucleus. So it's the thalamus, where we have many synaptic contacts between the neurons, and from this medial geniculate nucleus, thalamocortical neurons are produced, and their axons they reach primary auditory cortex in the temporal part of the brain cortex. This is the central part of the acoustic analyzer. And at last, vestibular analyzer. Peripheral part of that analyzer is located at the vestibular organ, that is also in the temporal bone of the skull. We have here semicircular ducts. We have 3 semicircular ducts in dry-dimensional world to be aware of the position of the head in the space, and to analyze the velocity and the angle of movement if the head produces the movements. And these semicircular ducts are filled with special fluid, endolymph, that contains small particles, otoliths, the special small stones that stimulate the mechanoreceptors in the semicircular ducts, and when the position of the body of the head changes, these small stones produce the stimulation of mechanoreceptors that then may be conducted through vestibular nerve to the brain, to vestibular nuclei, and then this information is propagating through vestibular spinal tract to the spinal cord, also to cerebellum and to the cortex, brain cortex upward, because we may be